So this is what I was most afraid of, was that
everyone else was going to be a lot scarier. And I'm pretty sure that's what happened.
So I thought about maybe bringing my pet velociraptor named Toby to scare you guys, but I haven't hung out with him since I was 7 and he was invisible. So instead, we're gonna maybe scare you with the pace of information that we're gonna go through. So let's get started. So in the last 100 years, we've split the atom. We fused it again because we got bored. We put people on the moon, we found the structure of DNA, that's pretty cool. And we used an international super collider to find the Higgs boson. We're pretty cool. But that begs the question of why I can't show you a real Toby. Or a tribble, just a ball of fur that makes noise, or have an eyeball in my knee, like that song. So what's going on? That should give you a sense, given the previous accomplishments we've had, of how difficult this problem is. So we're going to look at why it's so difficult in 10 minutes, and look at some new things that are coming out in different ways of thinking about how to grow biological structures. So we start with where this story began, which oddly was in the 1500s in Italy. So put yourself back there and assume that you're going to lose part of your nose to either syphilis or a duel. You can take your pick. I would pick the duel. So this was a big enough problem that 2 surgeons saw a market opportunity and they said, let's fix the nose. And so they would take patients in with an injured nose. They would form a scaffold out of cotton fibers that would reshape the part of the missing nose. And they would peel skin back from your bicep, but they'd leave it attached, and they'd strap your arm to your head and suture this flap of skin over your nose. And a couple weeks later, hopefully it had healed. There's little scissors, snip, 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 bada bing, bada bang, you've got a nose. And we have letters attesting to the efficacy of this technique. We actually still do something quite similar today, although the skin comes from somewhere else. And the moral of this story, though, is that from the very beginning, our work with tissues was clinical. It was about building repair kits and new pieces of the body, but it wasn't about building individual structures. And if you fast forward 400 years or so, you get to the birth of modern tissue engineering, which came from Langer and Vacanti in Boston. This was the original picture of cells being merged with a synthetic scaffold. So you take polymers, you make some shape, let's say a sheet or a tube. You put cells on it, you put them together, they grow, and you get some sort of tissue structure. Now, 10 years after that, we get the Vacanti mouse, and this is maybe the closest thing in tissue engineering to a monster. You have a immunocompromised mouse, you have a scaffold shaped like a human ear, and you've seeded cow cells into this. So this thing is all kinds of confused. And the point of this, though, is that it really highlights this idea that in tissue engineering, the central dogma now is we use the body to help us as its own bioreactor. So biology is very complicated. There are thermal signals, electrical signals, mechanical signals, chemical signals. We don't really understand those. And on a clinical level, if we can make something where we make a shape and we put some cells on it and we stick it in the body and we let the body do the rest of the work, that's a great strategy. So it's not a great engineering strategy, but it helps people. And we've done some nice things with that. So just a couple of those. In the upper left, you can get a custom bladder with your own cells in it from Anthony Attella's lab in Wake Forest. This means you can get it bigger than the one you have. So at that next local sports game or World of Warcraft session, you don't have to get up. And if you do have to get up, you can just get a tissue engineered urethra so your flow rate's faster so you miss less of the game. So this is actually quite remarkable. This is a full organ system that's been engineered from scratch outside of the body. That's very rare and quite impressive. And down here is the trachea. We just hit the 5-year anniversary of the tissue-engineered trachea being implanted in people. It's a 3D printed scaffold. You see cells on it, and you can replace parts of the trachea. These are very important and difficult and should not be underestimated, but they aren't standalone creatures or individuals. And that's an interesting point because everything in tissue engineering has been about building something that we know is possible to build. That's the existential problem in tissue engineering. What can and can't you build with a given set of cells? We don't really understand tissue as a medium. So we know that we can build a blood vessel because we can see them inside of us. But we don't know what else we could do. So we've sort of restricted ourselves in this area, and also it's just downright useful. But the problem is, if we wanted to go more in terms of what can and can't we build and how do we build things better, we come up against how the body does things. So we're completely outclassed. This is the early stage of development in a fruit fly within a couple hours. And what you're watching are three processes. The first is the synchronized division of these red dots, which are clusters of genetic material. They're not quite yet cells. The second is this bright orange or yellow patch that appears, which is cells performing a form of biocomputation, figuring out where to divide the line between, say, the thorax and the top of the body. And the last is this complicated collective migration of these things, all synchronized. That's how nature builds things. We build things more like this. And this is great. So I'm a mechanical engineer by training, and this is how you get trained. In any discipline of engineering other than tissue engineering, you have rules and standards. There are building codes, IEEE circuit designs, mechanical tests, and you can put everything together in a manual. You can teach that. There's a lot of creativity, but there are standard tools, and it comes in a lot of colors. In tissue engineering, we've got the colors down, but that's about it. There isn't a lot of standardization. Everyone has their favorite kind of tissue to work with, their favorite scaffold materials, their favorite applications. It's very hard to compare. And yes, it's a much harder problem, but we don't really have this mindset of the engineering practices yet. So a couple labs have started looking at this, like the Parker lab in MIT, where they made a jellyfish out of rubber and seeded it with muscle cells, and it swam around. And that's a little weird and maybe goofy, but the point is they used engineering principles to figure out how to do that. And it's a first step. It's this idea of introducing rational engineering to building a tissue. So that got people thinking, and people have been looking at new ways of thinking about what it means to be a tissue. So just like George was talking about, swarming is an interesting phenomenon, and it happens in things besides zombies. Just like spontaneous traffic jams, the mosh pit dynamics at a concert, flocks of birds, schools of fish, and the herding of sheep. And it also happens in healing tissue and moving tissue. So these are epithelial cells, very simple cell type. They like to spread out and form a sheet. And when you cut that sheet, they don't like this free space. So naturally, they heal themselves. And this takes about 12 hours, and it always happens. This is a very complex behavior that comes out of these very simple rules, and that's the nature of collective behavior. Lots of individuals together in a community interacting with simple rules result in these complex global patterns. So because this behavior happens, and because it's similar to the herding of sheep, I'm not going to lie, part of my dissertation was inspired by this immortal pig. And so it's Bahram you all the way, for those of you who aren't too young. So I thought maybe I could make a sheepdog that would herd tissue. It's a different way of thinking about how to manipulate tissue. It takes advantage of their collective behaviors. So the first sort of sheepdog I went after was this idea of using electricity. So back in the 1800s, this famous scientist came along. It's Emile Dubois-Reymond. He's an electrophysiologist known for lots of things with bioelectricity and for experimenting shirtless. That's true. And he also incidentally cut himself one day and measured a small current at the site of the injury. That's known as the wound current. And it took us about 100 years to figure out what that did, but when we figured it out, it's pretty neat. So first, why do we get an electric field when we get injured? So the answer is that we're very similar to the internet. We're a collection of tubes, except in our case, we're full of salt water. So when you damage those tubes, you get an electrochemical imbalance, you get a current, and the cells can sense that. So we think the model is that little receptors in the surface of the cell gently get pulled by this current to one side of the cell, and the cell knows which way to go. And this helps them in the healing process figure out how to navigate. So I asked, we've done this in single cells, can we do this in a tissue? Can we put a large group of cells together that are all chemically holding hands? That's what makes a tissue. It's linked together, and give it a signal and herd it. So this was one of the very simple early examples where you just have a field moving up and down, and you can make the cells follow the direction of an electric current. And you can do this with much more complicated currents and fields that go like this, or just split and things like that. And so what we're trying to do now is make a Band-Aid that takes advantage of this, and it sits on an injury, and it monitors the electrical impulses inside an injury and figures out, hopefully someday, how to stimulate back in to maybe heal faster or reduce scarring. And that was my dissertation work. I talked a lot about that in other times, and I'm working on something different now that's actually quite related. So let's say you want to take the sheepdog idea farther. Can you turn it into something where you make a physical sheepdog? So I'm hoping the answer is yes, because there's a lot of things you could do if you can pull that off. So these are my first sheepdogs. They're not furry yet, but they'll get there. So I used a micro silkscreen process essentially to make these little blocks of polymer. And eventually I'd like to coat them with a protein that cells use to recognize other cells. So now you have this idea that you can make a material and you can coat it in a way that it infiltrates an existing tissue and is accepted by that tissue as being tissue. That's different from current scaffolds, which are encapsulated or grown on by cells, but they're not treated as being part of the tissue in the same way. So it'd be neat if we could pull that off. So I made some early attempts without the protein. These are just the controls. So we have kidney cells here playing around with one of these particles. Nothing too exciting. They push it around, and eventually, if we let them grow long enough, they'll wind up underneath it, and you wind up with a big sheet of tissue and these particles sitting on top. What they then do makes absolutely no sense, but is fun to watch. So these are the world's smallest, most expensive beach balls. And this, we have no idea why this happens. But, so you get these interesting behaviors anyway. Hopefully when we put the protein on, the effect will happen. And it's nice because you get to work with artists and ant colony specialists, physicists, and all sorts of people when you look at collective things. So it'd be great to get some input from anybody here. And lastly, I did try and make a monster for you guys. Her name's Tiny Tops. She's the smallest living dinosaur-shaped object. These dots are individual nuclei of kidney cells. Yes, I could have made a raptor, it's true, but if you've watched Jurassic Park, there's a fundamental lesson when you're making monsters. You don't want that water drop or ripple thing to happen. Do not start with a Metasaurus when you're making a monster. So, thanks.